Garby, garby, gar. Morning, everybody. Would you want trouble? Come on. Come. Up. There we go. A dog sitting on the neighbor's dog. So now we have 3 dogs and 3 cats. Yeah, even number. All female too, so I'm even more outnumbered than usual. Guest dog professor, that's right. Yeah, haven't had too much sleep for the last couple of days, so hopefully I can hold it together until the end today. Hence the second cup of coffee this morning. All right, so we left off going through the principles of 3-point crosses. Uh-oh, here's the other one. Let's get the chat back up. Get all my stuff prepared. All right, and so just to recap, let's bring up this scary looking thing. All right. What can you not assume when I give you a 3-point cross problem to work through? It's not that I'm lying, you just can't make this assumption. Right, that the gene order is correct. Okay, that is something that you have to work out for yourself. All right, so how do you work out the gene order? What's going to tell you what order the genes are in? I mean, you could just guess, but that'd be pretty risky. Nope, not the distance. That's kind of the last thing you do, actually, or more or less. That's step 3 and 4 of the 5-point patent pending plan for success. What tells you the gene order? Let's get back up to here, give you a little reminder. So, yeah, I mean, kind of, definitely. I mean, that's the point of a 3-point cross, Adam, without a doubt. But what bit of information in this 3-point cross, right? If I bring this one back up. All right, what bit of information are you looking for here? Come on, you got to remember the 5-point plan. Okay, give you a hint. In this example here, in which of the A, B, or C, no, not quite. Which of the A, B, or C is the thing in the middle moving? Right, Cameron, you want the one with the 2 lowest recombinants because those are what? What individuals are the rarest in a 3-point cross? You got it right, Cameron. As the lowest values for sure. Well, you have 4 different classes of individuals. Yeah, Maria, that's the one. Double crossovers. So double crossovers are the rarest, like these ones here on the right-hand side, because you have to have 2 independent crossover events one between the first two genes and the other between the second two genes, right? So probability of independent events is multiplied, right? So it's even smaller, right? So those are the rarest ones. And the only way you can get B moving in this one here in a double recombinant is if it is in the middle. So just type that up again. Yeah, that was a really important piece of information, Rose. Sorry, I'm not laughing at you, I'm laughing with you, promise. Yeah, so double crossovers, which are the rarest, tells you the gene order, as it's the one in the middle that's moving. Okay. Does that make sense to everybody? If it doesn't, pipe up, because we need to get that straight first. And honestly, you might not believe me, but this is really, they're really easy to work out, as long as you follow the rules. That's the key. Okay, so that's all straight. Now what do we do? Okay, we've found the double crossovers, because they're the rarest. We've figured out gene order. What's step 3 and 4? I need to get my wife to make me a t-shirt with this on it. That'd be fun, actually. How to work out double cross, 3-point crosses. She did this one for me for Father's Day, which I thought was pretty cool. Yes, exactly, Rose. Figure out the single crossovers, and how do you do that? Could I make you think? Yeah. So you look for reciprocals. Good. So you found those. So you kind of group those 8 lots of different phenotypes into the 4 groups: parentals, single crossovers for one gene pair, single crossovers for another gene pair, and double crossovers. Cool. So you got that information. What do you do next? Yes, but how? How do you work out the recombination frequency? The reason why I'm badgering you about this is because I want to make sure you understand the bits that you need to include. If you remember back to the downsides of doing a bunch of 2-point crosses, what events do you miss if you did 3 2-point crosses instead of 1 3-point cross? They also happen to be the rarest. Yeah, that is exactly it, Rose. Thank you. Right. So to work out recombination frequency, 1, you need the single crossovers, right? The reciprocals that Cameron and co mentioned earlier. And 2, you need the double crossovers. You add those together and divide it by the total. Cool. Thank you, Rose. And then you do the same for the second gene pair, right? Which is the same deal, basically. Different set of reciprocals for the single crossover. And then the last thing is draw the map. Okay, anybody got any questions? And we'll go on to an example. Again, memorize those rules. Follow them. You cannot get this wrong. This is one of the joys about this type of genetics. Okay, everybody ready to go on to the example or the practice? Right, I've got two for you today. One on the PowerPoint and the other one which I actually worked out for you, which is actually, I mean, they're all based on real life examples, but this is based on one that we've actually seen part of before. Okay, so here's our experiment. They've even worked out the total for you. We have 8 different groups of phenotypes, right? And so you've got the essentially the genotype here and the phenotype here, right? And we've got the number. All of these have a triple recessive kind of chromosome as a kind of a tester chromosome, right? So the bottom chromosome in all of these is recessive for all 3 genes, right? Because it's a kind of a back cross test. It's how you do these things. The other chromosome, the one at the top, is what you see the genotype for here. Okay, so which ones are the double crossovers? Oh, can you zoom in? Sure. No, don't want to zoom in on that. Not that much. That better? Cool. Okay, so step 1. Find the double crossovers. Which ones are those? Come on, peeps. Spineless, scarlet, and ebony. Oh, spineless is spineless, and then scarlet, ebony. Correct. Good stuff. Haha. See, I'm being encouraging. I've been told to, you know, encourage people and stuff. Okay, so what's the gene order? So, hint, look at the parentals. So if each parental, which are the ones with the largest number, they're really obvious, and then put the top there for you, which is rather nice. One parental is all wild type, one parental is all mutant. Okay, so you're looking for what has moved between these two to create the double crossover. So which gene or allele has moved? It's actually easiest looking at the single mutant double crossover because that's the one that's moved, right? So which one has moved? Yeah, SS. Good job, Valeria. That's exactly right. And then you can double check that with the other double crossover, right? So single crossover in spineless, it's the SS, right? The other two are wild type. And in Scarlet Ebony, the thing which is normal is also SS, right? Because they're reciprocals. So SS has to be in the middle. So what's the gene order? Looks good to me. Or, you know, if you want to be contrary, could be ESSST. Either's correct. I'd go with the same format that they have on here because it just keeps things a little bit simpler, right? So gene order is STSSSE. So step 3, RF between ST and SS. Not SSN, I don't need social security number, I meant SS. Okay, so you're looking for, we've got the double recombinants already. Now we need to find the result of a single crossover between ST and SS. Right, so what's that going to look like? So let's start with ST. We're going to have ST plus. What are the other two alleles going to be if the crossover is between ST and SS? So again, if we're crossing over between ST and SS, we're going to lose the wild type SS allele and the wild type E allele. So what flies look like that? They're going to be wild type for ST, recessive for SS and E. That's right. Good job. Then we look for the reciprocal. Right. Those two are the result of recombination between ST and SS. Ooh, hang on, let me double check, make sure I got this right. So we're going to have a wild type ST, recessive E and SS. Then we're going to have a recessive ST, wild type E, and wild type SS. Yeah, good job. Pat, pat, pat. All right, so you have all the information you need. What's the recombination frequency? And please, if anybody's lost, right, and you're like, what the hell's he talking about? Ask, please, because I'm not going to be able to figure out what you don't understand if you don't let me know that you don't understand it. So what numbers are we looking at here? Okay. All right. So let me draw this up. It's always easier to draw something than it is to explain it with words. I'm just going to, can you see this big enough? Chew toys out of the way. No, need to stop the share. Was that a head shake, Robert?
No, you're good. I can see that.
Is it big enough? Good. Okay. So, yeah. We have ST plus, SS plus. You know what I'm actually going to do? I'm just going to get rid of and just do plus. The way they've done it is correct. I just think it's a little simpler to look at it like this. Okay, so this is a cross. This is the correct gene order that we have. Right, so essentially, if we're going to have a crossover here, right, so we're looking for single crossovers between, and hopefully this will answer your question too, Adam. We're looking at single crossovers between ST and SS. We're going to have, and you can just follow it as well, like with your pen, plus SSE. Which is the ebony spineless, right, that Rose picked up. And we'll have the reciprocal, right? So we'll have ST plus plus. And if you look at that, that is, where we're at, ST negative plus plus, that's the scarlet ones. So scarlet, that's 52. Ebony spineless is 50, right? So these are the reciprocals for the single crossover, right? But if you want to work out recombination frequency, you can't just use these, because the recombination frequency also includes double crossover. Yeah, double crossovers. Right, so we want these ones too. Because we don't care about the crossover between SS and E. We do care about the crossover between ST and SS in the double crossovers. So that's why they go into the calculations. And those will be plus SS plus ST plus and E, right? And that is, which one has plus SS minus, that's 5. And the other one is 3. Okay. Oh, Adam, so I got the 50. So, where's my red pen? If we're looking at a crossover between these two genes, that's what the X is all about, right, Adam? So if we have a crossover, essentially you're going to be adding this bit of this chromosome to this bit of this chromosome here, and vice versa, opposite too. So if you draw it out, you're going to have plus SSE. Make sense? Because this bit of one chromosome, and let's draw it up with 2 lines. And let's do different colors too. Don't worry about changing this on your own stuff. This is just, let's draw it with 2 lines, right? Same information, maybe a little bit clearer. Oh, wrong colors. Where'd my, there we go. Okay, so two different chromosomes. We're going to have a crossover event between the two chromosomes. These are, again, the non-sister homologous chromatids, right? So if we have a crossover between these two genes here, DNA gets broken and then stuck back like so. That is going to give you this red bit and this blue bit like so. And because crossover is always reciprocals, you're going to have the opposite. You're going to have ST and normal versions of the other two. Now you take this and you go look at your screen, right? And you go, okay, if I've got normal for these two genes and I've got a recessive allele for this, right? The ST stands for scarlet. I'm going to look for the ones which are scarlet. And you look down in this table here in front of you, we've got 52 of those. Now you come back to your board and it's usually fairly obvious which are the reciprocals, right? Because they have pretty similar numbers as you'd expect. But even if not, you can double check and look at this and go, okay, scarlet, what's the opposite of scarlet? Well, it's going to have two mutants, two mutant phenotypes, because it's going to have, instead of just scarlet and plus plus, it's going to have plus and what's that? Spineless and ebony. Okay, so then you go look for spineless and ebony, and that's the 50. So these are single crossovers. These are our double crossovers. How about that? Did that make more sense? Good. Okay, so to work out recombination frequency, we need the single crossovers and the double crossovers. We add them together, which gets us, what's that, 102 plus 8, so that's going to be 110. Just so you can see down at the bottom. Playing dog's toys and stuff. So recombination frequency equals that over our total, which is 755. And that's hiding behind the chat box now. Yep, give me just a second, Valeria. I'm one hand calculating. Okay, times that by 100 and run out of space. That is 14.6. Most important value is stuffed in the corner of the whiteboard. You'll see when I take a picture of it. Okay, so Valeria, the double crossovers are the rarest events. They're the rarest events because you have to have two independent events. hang on, Adam, explaining something to Valeria. Right, so you have the probability of having a crossover here times the probability of having a crossover here. That will give you the double crossovers, right? So they're always the ones with the smallest numbers. Without exception. And so you look for the pair of reciprocals that have the smallest numbers. And in this case, they are that one and that one. Those are the double crossovers. Did that make sense, Valeria? Cool, good stuff. Adam, not quite sure what you mean by the question, but it is definitely a process because you have to follow the rules and you'll get it right. Okay, so someone make a note of that number, 14.6 centimorgans. You figured it out? Good stuff. Okay, so now if we want to figure out the crossover here, let me just take a picture of that. Oh, I can do the obligatory dog of the day picture. That's Snowball, by the way. All right. So let's just put this back how it was. Okay, so now we have crossover here. South African. Now we generate plus plus E, which is going to be ebony. And the reciprocal to that is going to be scarlet, spineless, and that's it. So you look down through there, look for ebony is 43. Scarlet spineless is 41. I've handily grouped them together for you, but it wouldn't hurt to just double check that yourself. And so then our total is going to be 84 plus 8 is 92. Oh, yes, that's correct. Sorry, Rose. This is between SS and E. Now we have 92 divided by 5, and then we're just going to times that. Fingers don't stretch that far. Times 100. That is now 12.2 centimorgans. Down there in the corner. Should have left myself more space. Okay, so those are recombination frequencies for those different genes. And then the last step is simply to put in the distances. And you can do this as a double, you can do a single, it really doesn't matter like how it looks. This is going to be 12.2 and what was it, 14.6, I think. Correct me if I'm wrong. And there you go. That's it. Nothing more to it than that. Okay, got one more practice. Yes, that's correct. But if I'm being a pedant, Adam, it's centimorgan like that. If it's both lowercase, people think it's centimeters, and that's obviously not right. Okay, questions? Anybody? Questions? When you think of that, I'm going to write up the next one. Okay. Oh, and also, Rose, you asked about checking your answers in the exam one. I found out it's because the exam had disappeared from Student View. So I've made it kind of available again. And so you should be able to see it. So have a look. If you can't find it or it doesn't show you what you're looking for, let me know and I'll try and figure it out. But yeah, hopefully you should be able to see it now. Okay. Let me tip this round. All right, so this is actually a 3-point cross that I worked out from actual chromosome data. And it's part of it you've already seen kind of before earlier in one of the 2-point crosses examples. So this is the cross we're doing, standard back cross between a individual that's heterozygous for these 3 genes and a recessive male. Double quick check. What linkage phase are we looking at here? That's in cis. Good stuff. Okay, so now we have, I'm not going to write out the phenotypes because that would just take ages and it's not necessary. But I'm going to write out the different, because genotypes equals phenotypes essentially in this case. And this will go up as a picture too on Blackboard. So don't get too stressed out about copying it down. Gotta make sure I write everything down, otherwise it'll be really confusing. All right, here are the numbers. Okay. There we go. Can you all see that clearly? Those are those are M's, and this is a W. All right. got a little bit of time to get their head straight. Good questions, everybody. Rose, there will only ever be one double crossover. So you can have genes that are very, very close together, in which case it would be even less likely to find double crossovers. But yeah, you'll only ever get one double crossover. And Adam, yeah, these were worked out from actual, the numbers were worked out from actual chromosome data. So I went on Wormbase and looked up some genes and got their linkage data from that, not Wormbase, Flybase, sorry, and then worked out the numbers from that. So I'm going to give you a few more. It seems like people are working it out themselves, which is cool. And I'm going to let people have a few more minutes to do that before we go through it all together. So we have W, M, and DM are the genes. Like so. Oh yeah, and those are pluses. Okay. People still working it out, or should we go through it? Still working? Okay. All right, let's work through this problem. So first things first, let me put my overhead light actually, that might help. A bit of shadow from the laptop and the front window. Okay, so and this is an example of the kind of thing that you'll get in the exam as well, right? So practice these. You want to be quick. You don't want to spend in 15 minutes on just one question. Okay, so first job, which ones are the double crossovers? Are they these ones? WM, or let's work with the nomenclature that's on here, Adam. You're correct, but we'll just avoid confusion. So WM plus DM. These are our actually, let's just call them what they are. They're double crossovers. Okay, so what's the gene order? Which thing moved? If that was the gene order rows, would a double crossover result in a, well, what would a double crossover result in? If we have cis linkage at the top. You would end up with, it would be, if that were the correct one, now let me just turn this around. That would be plus W plus and M plus DM. So we need to look for a plus and M and DM. That's here, and W plus plus, yeah, or plus W plus would be here. These are not the double crossovers. Even though their numbers are similar to these, they are not the lowest numbers. So good shot, but no cigar. So just to illustrate that further, that's a single crossover. And this is a single crossover. So what's the correct gene order? Which thing has moved here? Yeah, that's exactly right. Thank you, Maria. So this is actually incorrect. It is WDMM or MDMW, doesn't really matter. Okay, so what's the distance between W and DM? Which of these single crossovers results from a crossover event between W and DM? So basically you're looking for W on its own. Actually, let's number these. Yeah, exactly. Because we have W on its own, plus plus, and the reciprocal is going to be wild type on its own, or plus on its own, MDM. Right, so basically it's going to be that, oops, can't see. Yeah, there we go. That normal, or those normal, right? That's what we've got here. So recombination frequency is, how many of these do we have? 6 and 4. How many of these do we have? We've got to include the double crossovers. 3 and 2. What's the total? I haven't actually added it up. Someone can do that for me. $980. Thank you, Rose. Then times all that by 100. And that is recombination frequency WDM. So that's what, 15 over 980 times 100. What's the centimorgans, or the recombination percent? $1.53 centimorgans. I think, Adam, you forgot to times it by 100. Okay. Okay. So combination frequency between DM and M equals what? Okay, can you take me through your workings, Rose? What numbers are we using now? All right, so now we have single crossover here, single crossover number 2. Plus our double crossovers. Again, over 980. And then times that all by 100. Just see there. Right, and that gives us second crossover is between DM and M, Adam. And so we still need the double crossovers, the 3 and the 2 from here. Now we need the data from our other single crossover event, which is this one. Add those together, divided by the total, times the whole lot by 100, and you get, we're just going to round up to 1 decimal place, 33.7 centimeters. Centimorgans. I'm saying it myself now. So really, this looks like so. This is going to be 1.5, and this is going to be 33.7 centimorgans. That's not a problem, Valeria. That's awesome to know, because that's how we figure out what's what. So for the gene order, what you want to know is which one of these things moves in the middle, right? Because the one that moves in the middle is a result of a crossover event here and here. Right? You have to figure out which one in the middle is moving, right? Because that is the one that is produced by having a crossover here. Basically, you do this, like that. And then the opposite, like so. So you want to know which one is in the middle in a double crossover. And so double crossovers are the rarest. So you find the rarest individuals, ones which occur least frequently, which even though we have two groups that are pretty low in number, this group has a lowest number, right? So that tells us that this group is the single crossover, the double crossover group. This group is a single crossover. So from the double crossovers, now we look here and we go, okay, which one is moving? The one that's moving is going to be the one, like the odd one out, essentially, which is going to be this one and this one. They're the odd ones out because if you look back up here at the, or even if you just look at the parentals, right? Here's parental 1, parental 2. So we have plus, plus, plus. What you're going to be looking for in a double crossover is a plus, plus something, right? A single mutant allele. Yep, that's exactly how you do it, Rose. Double crossovers, look for the odd one out compared to the parentals, right? You always have to look at the parentals first. That's their sole use, right, in this process. And see which one has moved, right? If the one that moves is W, W's in the middle. If the one that moves is M, M is in the middle. If the one that moves is DM, DM is in the middle. Now we go down here and look at our double crossovers and we find out it's DM is the one that has moved. So DM has to be in the middle. It's the only way it could get swapped out and the other two stay the same. How was that, Valeria? Did that help? That make sense?
Yeah, so essentially we're always going to compare the double crossovers to the parentals.
Correct.
Okay. All right. I think I get it a little bit more.
Cool. But the, you know, I kind of joke about like my patented pathways to success or steps to success. And that's really the reason why I kind of bang on about those is that if you follow those rules, those steps, you'll always get it worked out just fine. But you have to start with the double crossovers to figure out gene order. All right, everybody, time to end this one. Stretch our legs and start the next one in, I'm going to try for 1/4 just so we don't run late on the lab. But if it takes a little bit longer than expected, that's fine too. So going to aim to be back here at 12:15. Okay. If you still have questions about this stuff, write them down, hold them in your head, whatever, and ask them when we come back. And then we'll move on to DNA structure and DNA replication. Cool beans. See you in a little bit.